Hello, welcome back to the Computational Genomics Lab. Today we're going to do one of my favorite things is look at raw data. So we've been making all these wonderful figures and plots, heat maps, but those values are kind of mysterious. And when you can actually see the count stacking up across a gene and changing, that's when you're really going to trust it. So you do typically your DE-seq2, find genes that are really significant, look at the raw data, and then start making those plots because you now trust it because you've seen the raw data. And the amount of time I spend looking at raw data, and I love it, but is a lot. And I think it's really important to dig into that. And so that is the goal for today's lesson. We are at the end of level 6 and ready to level up, level 7 visualization, and we're ready to level up to level 8, which is going to be AttackSeq, which we would do the same thing and look at the raw data of AttackSeq. There's no type of genomic analysis where you don't want to look at the raw data. And so that's what we're going to do today, and it's actually quite simple and easy. So I have everything outlined here for you, but we'll just walk through this together to look back at our NF Core pipeline output gave us some really cool files we can use to look at the raw data. Okay, so first step is download IGV. So you're going to take that web link I have pasted in there and look for whether you want it on Windows or Mac or Linux, but make sure you get the Java included version. You want the Java included version, or you will be tracking different versions of Java and you might mess up previous stuff on your computer. So I'm not going to download it because I already have it downloaded, but click your button, it'll download, standard opening of the file. And then, that's just saying pick the Java version. And that's really the main crux of it. So this is what IGV is going to look like when you open it up the first time. You're going to want to make sure you're on this mouse MM10. And that's really important because as genome versions change, the positions and coordinates change, and the raw data is just going to tell you the position of the read in the genome. So if the genome shifted by a million bases, that one read in one genome is going to be a million bases in a different place. And can really throw you off. So MM10, you can pick other versions of genomes, chickens, zebrafishes, there's C. elegans, all kinds of different genomes you can use. But then now what we need to do is go to a file transfer program. I'm using CyberDuck here. You could be using FileZilla. And in 04, we did RNAseq. We actually ran the pipeline there. And so I'm going to where my pipeline output was in the results here. And if I look in this star salmon folder, I see a folder called BigWig. And that stands for wiggle files, but really it's a summary of how many reads landed at each position in the genome, or the raw data, if you will. The rawest data is those FASTQ files we put into the pipeline, but then this is sort of raw counts of it afterwards. And we have 15 files, 3 replicates for 5 different time points. This is probably going to take, it's going to be a few gigs of space, so make sure you have that on your local computer. But you would just drag this down and transfer it. But I've already done that because it took, I think, over an hour. But then watch how cool this is. So we can just highlight these, drag them right into IGV, and we can start to explore our raw data. So I keep mentioning this H19 gene. Again, I like long non-coding RNAs, and H19 was the first long non-coding ever discovered. But in this box here, you can type in your favorite gene, and man, it doesn't look like there's any data. How could that possibly be? That might be concerning us. But there's a lot we can do over here because the IGV is going to scale them individually. And so if we highlight all of our samples and then right-click or control-click on a Mac, you can do group auto-scale. So it's going to say, take all of these things and scale them to the same level across all the samples, whereas right now they all have different levels. And voila, we can start to see our data. We can zoom in a little more by just scrolling up above on here. I'll make this a little bigger. And IGV is really fast. It uses Google's caching algorithm so that there's tons of data being thought about, but it only focuses on the screen you have in front of you. So it will get slower as you zoom out, but you can zoom in manually by just going up here and see this blue box saying where we want to go. So we zoomed in a little bit. And so, we forgot to auto-scale the first two samples. You can see that this red is telling you that they've been auto-scaled. And now let's make sure we get all of them auto-scaled, group, auto-scale. Cool. So what we can see here is they're ordered by time. So as we go down here, we have more and more time. And we can see H19 is like the perfect example of a gene that is being regulated by DOX in the mouse stem cells. And so we can see it's low, 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 and then it gets really high around 96 hours. It really seems to change around 48 hours. Again, a pattern we've started to see emerge in our data. And now that these are auto-scaled, you can start to go look around the genome. Let's see, what's a gene? Actin. Okay, we go, oh, it doesn't, I didn't spell it right. Let's do HOX A13, another one of my favorite genes. Doesn't look so great. And you can see that that was probably not one of our genes, but that's where you can tell the difference between good data and bad data. There's also some things a lot more things you can do, and a lot of resources on IGV out there. You can also load different data sets, so there's different annotations here for mouse MM10. You could add repeat masker for transposons, but I like to leave it here with just the genes, so you can search for any of your favorite genes, or you can type in the position in here and go there. You can zoom in and out up here and start to look for things in the data. So, ooh, wow, this looks like a very abundant gene. And then we can just zoom in on it here. Boom. It's abundant in all of our samples, which means that's great. It looks the same, and it is not being affected by DOX. And so you can also just hold down and scroll across the genome. I find this really fun. It's like a video game for genomics, and get to know the genome, but then you can see all of your data group scaled in here. And so when you've done your DE-seq analyses, and I encourage you to take some genes with high log fold changes, significant p-values, and just take a look and browse through and see who looks good, who doesn't look so good, because just because the statistics were there doesn't mean the raw data actually looks that great. So congratulations, you've finished all of your visualization exercises and can make heat maps, line plots. You know the difference between TPM and count plots. And now we are ready to end our RNA-seq journey. And I think you're at a really good place knowing some basic R fundamentals, where you can start to use ChatGPT to type in some code and say, hey, now I want to do this. Can you help me do that? I find it's a really helpful resource, but you have to know the fundamentals first. And you're there, you know how to look at the raw data and the genome, and we're good to move on and level up to 8 and start doing ATAC-seq. All right, we'll see you there. Have fun browsing the genome.